आज हामी एप्लिकेसन अफ जुलोजी च्याप्टर डिस्कस गर्न गइरहेको छौँ। यस अन्तर्गत विभिन्न टपिकहरू आज हामी एप्लिकेसन अफ जुलोजी च्याप्टर अन्तर्गत विभिन्न टपिकहरूको बारेमा डिस्कस गर्न गइरहेको छौँ। त्यसमध्ये द फर्स्ट टपिक इज अर्गन ट्रान्सप्लान्टेसन। अर्गन ट्रान्सप्लान्टेसन भनेको यदि कुनै मान्छेमा कुनै अर्गन चाहिँ काम गर्न छोड्यो भने त्यो मान्छे बिरामी हुन्छ अनि उसको अर्गनमा चाहिँ आज हामी एप्लिकेसन अफ जुलोजी च्याप्टर अन्तर्गत विभिन्न टपिकहरू डिस्कस गर्न गइरहेका छौँ। त्यसमध्ये सबैभन्दा पहिलो टपिक अर्गन ट्रान्सप्लान्टेसनको बारेमा आज डिस्कस गर्दैछौँ। अर्गन ट्रान्सप्लान्टेसन भनेको नेपालीमा अङ्ग प्रत्यारोपण भनिन्छ। अहिले हाल नेपालमा पनि विभिन्न अस्पतालहरूले स्पेसली किडनी र लिभरको चाहिँ ट्रान्सप्लान्ट गरिरहेको हामीले देख्न गरेका छौँ। र यसमा के हुन्छ भने कुनै मान्छेको चाहिँ अर्गनहरू काम गर्न छोड्न सक्छ। अनि त्यो कन्डिसनमा त्यो मान्छेले अब कुनै हेल्दी अर्गन चाहिँ आफ्नो बडीमा राख्नुपर्ने हुनसक्छ। त्यो कन्डिसनमा अब डोनर जो चाहिँ हेल्दी छ उसले आफ्नो अर्गन चाहिँ बिरामी मान्छेको त्यो जुन अङ्ग काम गरिरहेको छैन त्यसको साटोमा चाहिँ दिने गर्छ। अनि दिस प्रोसेस इज कल्ड organ transplantation, where one donates the healthy organ and other receives healthy organ. And the person who donates organ, उसलाई हामी डोनर भन्छौँ भने the one who receives organ, healthy organ, उसलाई recipient भन्छौँ। That's why the process of replacing the recipient's damaged organ, जो यो बिरामी हो जसले रिसिभ गर्छ उसलाई रेसिपिएन्ट भन्छौँ, by donor's healthy organ is known as organ transplantation. र अर्गन ट्रान्सप्लान्टेसन चाहिँ कुन गर्न सक्छ भन्दाखेरि हामीले हार्ट, किडनी, लिभर, लङ, pancreas, intestine and skin. These are the organs which we can transplant from one person to another person. Along with that, we can also transplant tissues like bones, tendons, cornea, heart valves, and veins. And those tissues or organs which are transplanted, those organs or tissues, they are known as graft or transplant. And when we transplant, then the recipient body may behave those organs as a foreign organ or tissues, and because of that the recipient body may reject those organs. In that case, before we transplant, we have to go with the test like human lymphocyte antigen, or it is also called HLA test, or tissue typing. So after doing this test, if a result comes positive, then only the donor can donate its healthy organ to the recipient. And there are 4 types of organ transplantation that are in practice. The first is autograft or autogenic graft. Here the donor and recipient are the same person, is the same individual. So what happens? Some part of body is removed and transplanted into other part of the body within the same individual. Such transplantation is known as autograft. So since the person is same, so here there is no any chance of rejection of those organs. Like example, you can see skin grafting, vein extraction, hair transplantation. These are the examples. Now let us go to another type of transplantation. It is called isograft or syngenic graft. So here the donor and recipients are different individuals with identical gene. So where you will find identical genes is identical twins. So they have identical gene. That is why when organ transplantation is done between this donor and recipient, rejection is not a problem. I mean to say, there will be no rejection. The third is allograft, or it is also called allogenic graft. So here there are two different individuals of same species, and since they are two different individuals, so genetically they are different from each other. So in this case, what happens is before we do the transplantation, we have to have test like HLA test. If result comes positive, then only we can do transplantation process. In human, it is in practice, especially major organ like kidney and liver. Here, one person donates healthy kidney to the recipient, or the healthy person donates liver to the recipient. But as I told you, before the transplantation, both recipient and donor they must have tissue typing test or HLA test, and if the result comes positive, then only we can do the transplantation. The last type of transplantation is xenograft or heterograft. It is also known as xenogenic graft. Here, donor and recipients are from different species. Since they are different species, so such transplantation is extremely dangerous because there might be high chance of rejection of transplanted organ. So since it happens between different species, so the genes are totally different from each other. So that makes it high risk. So we can see the example pig heart bulb transplants. So here what happens is the bulbs from pig heart are taken out and transplanted into the heart of human. So how to prevent rejection problem? So there are some methods by which we can prevent rejection problem. The first is selection of organs from genetically similar member. So whenever we select donor and recipient, then if we do this process among the siblings, then what may happen is among the siblings there will be some kind of similar genetic composition, and that gives least chance of rejection of organs. Next is tissue typing. As I told you before we do transplantation, we must do the test like HLA test, and when the result comes positive, then only we can decide that transplantation can be done. The third is exposure by radiation. So here the main idea is by the radiation, the immune system of recipient is suppressed, and due to that what may happen is the received organ will not be rejected from the recipient body. Similarly, what we can do is we can take the drugs which can suppress the immune system of the recipient. So point number 3 and point number 4, the objective of point number 3 and point number 4 is same. In both methods, we suppress the immune system of recipient. In third, by radiation, and fourth, by taking the drug. And those radiations and drugs will suppress the immune system of recipient, due to which the transplanted organs will not be rejected. So this much about organ transplantation. Now let us go with the another topic, and the topics are amniocentesis and test tube baby. So first, let us discuss with amniocentesis. So amniocentesis, it is also known as amniotic fluid test. So the fluid that is present here, that fluid is called amniotic fluid, and its test is known as amniocentesis. And it is mainly done to diagnose diagnose chromosomal abnormalities and infections of fetal as well as gender discrimination of the fetus. In this procedure, the small amount of amniotic fluid is taken out with the help of syringe, and then its DNA is examined for any kind of genetic abnormalities, which helps to check either the fetus has genetic abnormal disease like Down's syndrome. Women who choose to have such tests are primarily those who are at increased risk for genetic and chromosomal problems because the test is invasive and it can carry risks for miscarriage. So that is why it is not done with all the women. It is only done in those women who have such risk. Similarly, this kind of test is done to find out the gender of fetus which is growing inside uterus of mother. So how to do this process? So it is done in about 14 or 15 weeks after conception by following methods. Firstly, ultrasonography is used to locate the position of fetus in the uterus. So where is the location of fetus? That is finalized using the method called ultrasonography. Next, using the long sterile hypodermic needle, the amniotic fluid is withdrawn from the amniotic sac. The third step is whenever the amniotic fluid is withdrawn, along with the amniotic fluid, some fetal cells are also withdrawn, which are placed into culture medium and stimulate them to grow and divide further. After that, the two types of tests are then performed on the cultured fetal cells. The first is karyotype test, where chromosomal abnormalities are detected. It also helps in determination of sex, gender, on the basis of presence or absence of sex chromatin. The second type of test is biochemical test, and it helps to detect prenatal disease such as phenylketonuria, sickle cell anemia. These diseases are uh, genetically abnormal diseases. So those diseases can be detected doing such test. So why women are taken for this test? The reason is if mother is over 35 years of age, then such test is done because when the woman is more than 35 years of age, then there can be other complexities. So to avoid that complexities, such test is done. Similarly, next is if the family history has some kind of chromosomal abnormality, abnormality, then to check the fetus, either the fetus has such problem or not. To check such disease, amniocentesis is done. Similarly, if the family history has some kind of inherited disorder, like disease, okay, genetic disease, then to check those disease in the fetus, such test is done. Similarly, the growing fetus, does the fetus has developed neural tube or not? To detect that, such test is done. However, there can be misuse of amniocentesis test. In general, this technique is used in society to know the gender of fetus, due to which there is increased number of abortion of normal female fetus, because in society everybody wants male child. So because of that, to know the gender beforehand, such tests are taking place. So this is the misuse of amniocentesis. Now let's talk about test tube baby. It is also called IVF technique, which is used if there is certain infertility problem in female or male. So test tube baby or IVF, it is also not known as in vitro fertilization. Here, fertilization of ovum by sperm is done outside the woman's body in the laboratory conditions. Applicable to this kind of technique is applicable to such women who are unable to conceive and give birth to child due to certain infertility problem in female and male. The world's first test tube baby that you can see here, and the name of that baby girl is Louise Joy Brown, and she was born on 25th July 1978. In Nepal, also we had the first test tube baby on 3rd March 2005, and his name is Om Tama. Now, how to do this kind of technique? So, process of IVF treatment. So, we go through series of stages. The first stage is the female is treated with hormones like follicle stimulating hormone and luteinizing hormone in order to make the oocytes matured. In second stage, a needle is inserted in the ovary to collect those matured oocytes. In third stage, the sperm is collected from the male. Now, after collection of both female gamete and male gamete, those are placed in sterile nutrient solution in order to fertilize, and those are incubated for overnight. In stage 5, we check either those oocytes are examined, those oocytes are fertilized or not. If the sperm is of poor quality, then oocytes may not be fertilized. In this case, the outer covering of oocyte, that is zona pellucida, the outer covering of egg, is damaged in order to make easier penetration of sperm, and finally the egg will be fertilized. After this, the mother, that mother can be surrogate mother or same female. In her body, the hormone progesterone is injected to prepare the uterus for implantation of that fertilize egg. And lastly, the embryo, which is 16 or 32 celled, those eggs are transferred into the uterus of the same female or surrogate mother using a small tube. This is how we can conduct IVF treatment. Now, it has certain drawbacks. So, let us discuss about that. The first drawback is the couple might have wanted only one or two children, but in this case, there can be several unwanted embryos are produced. So, because of that, the couple may have more than two children, and this technique is expensive, so normal people cannot afford it. So, as I told you, the several unwanted embryos are produced. That's why there can be multiple birth. So that can be twins or triplet, which can be risky for both mother and babies. Now the next topic that we are going to discuss is transgenic animal. So this is the animal where the foreign gene is introduced into their gene. So that animal is called transgenic animal where the foreign gene is introduced into their genome during the embryonic state. The first transgenic animal was mouse that was prepared by the scientist called Rudolf Ginesk in 1974. So there are some examples of transgenic animals which are really useful in human society. So the one example that I would like to discuss here is transgenic pigs. So transgenic pigs, it is used in organ transplantation without the risk of rejection. Just before we talk about the valve of pig is used to transplant in human heart. So using this technique, we can match the gene of pig and recipient. So in that case, the rejection chance can be reduced. Similarly, there are transgenic cows which are used to produce milk with protein. So it helps to fulfill the demand of protein in human. Similarly, there are some transgenic fishes and they are used to produce growth hormone, and those growth hormone can be used to treat dwarfism. Now the last topic of this chapter is poultry farming and fish farming. So let us discuss with poultry farming. So poultry farming means normally in Nepal it is understood as farming of chicken, but poultry farming is not only the farming of chicken, so it is farming of any kind of birds like chicken, duck, or turkey. And recently, some Nepalese they have started even rearing of ostrich. So these all are called poultry farming for what? Commercial production of eggs and meat for human consumption. So it has got certain economic importances. So few importances that we'll discuss here. It provides meat and egg in cheap price due to which the source of protein can be easily provided in the society. And the poultry droppings, they are the good manure. So mixing them in the soil, we can have the more crop production. And the feathers which are present on the body of birds that can be used to make sleeping bag, winter jacket, pillow, etc. And even it generates the employment that we call it self-employment, and with that we can generate the income. That is why Poultry farming is very famous in Nepal. Now let's go to another farming, that is fish farming. So here we rear fish at commercial scale for human consumption. And there are two types of fish farming. One is monoculture and polyculture. By the topic only you have understood mono means single. So if only single species of fish is raised, it is known as monoculture. And this kind of fish farming is easy because you have to just give one kind of food, and the since one kind of fish is kept there, so just providing one kind of feed is enough to the whole fish. But the risk is, since it has got only single species, so if a disease occurs, then the one single disease can kill all the fish in the pond. We have another type of fish farming, that is polyculture. So poly means many different species. So if many different species of fish are raised, it is called polyculture. So its production is higher than the polyculture because such in such farming, the different species of fish can utilize different source of food. And because of that, the productivity of farming will be more in comparison to monoculture. There are some fish farms in Nepal, mostly from the government side. So they are Godavari Fishery Center, it is in Lalitpur; Hetauda Fishery Center, Makanpur; Balajur Fishery Center, Kathmandu; Janakpur Fishery Center, Janakpur; Trishuli Fishery Center, Nuwakot; Dhangadi Fishery Center, Kailali; Pokhara Fishery Center, Kaski; Kulekhani Indra Sarovar Fishery Center, Makanpur; Rapti Fishery Center, Chitwan; Kulekhani, that we already said, and Bardia Fishery Center, Bardia, and lastly Bhairava Fishery Center, Rupandai. And commonly cultivated fish in Nepal that we will discuss here. So indigenous fish means those fish whose origin is in Nepal, and exotic fish means those fish which is brought from outside the country. So these are the some fish, name of fish which are cultivated in Nepal. Rohu fish. So this is rohu fish which is common in market. Naini fish, cutlet fish, salt fish. Similarly, those fish which are brought from outside the country, they are common carp, silver carp, grass carp, and big-headed carp. So now let us discuss of different types of methods of fish farming. So normally we have three different method of fish farming. One is extensive fish farming. So here the fishes are reared in natural environment without any external support. So in this case, there is no any input from the human. What we do is we simply release the fries of fish into the water. After that, it is left in natural environment. Since there is no any support from human, that is why the fish production will be low. Next we have is semi-conservative fish farming. So here there will be some human support, and since there is some human support, that's why it is called semi-conservative fish farming. And because of that, compared to extensive fish farming, there will be slightly more fish production. And the third method of fish farming is intensive fish farming. So in this kind of fish farming, there is high level of human support. I mean to say, there will be high involvement of human input. Because of that, the production of fish will be high because human provides care, they provide food. Because of that, the growth of the fish will be high. That is why production of the fish will be high in such type of fish farming. Now let us discuss about different types of fish farming pond. So the first is segregation pond. So since the name is segregation pond, so here we separate male and female fish before the breeding season. The second is breeding pond. So here breeding pond is a small pond, and here what we do is male and female fish, they are kept in the ratio 2:1. That means 2 part male and 1 part is female. And the main reason of keeping them together is for fertilization. Hatchery pond. So it is also a small pond where the fertilized eggs are transferred from the breeding pond, and those eggs are left in hatchery pond till they do not get hatched. Nursery pond. It is comparatively bigger pond where fries, the hatched, when the eggs are hatched, then the released small tiny fish are called fries. So those fries are shifted from hatchery pond into nursery pond and kept for 1 to 3 months till they become 10 to 40 gram in weight. And the last type of pond we have is rearing pond, where those small fish from the nursery pond are shifted and left for growth, and they are fed well till they become size of table fish, and once they become size of table fish they are sent to market. Now, to have the proper fish production we need to have proper pond management skills. So how to manage the pond? So some skills, some methods we'll be learning. The first thing that we have to take care is quality of soil. So the soil that is present in the pond, that bottom must be able to hold water, otherwise the water will not be able to collect in the pond. And similarly, the soil must be very much fertile because they need to provide nutrients to the aquatic plants, which are the food for the fish. Similarly, next is water. So water must be green in color because the green color means there is enough phytoplanktons and zooplanktons, which are the food of fish. Phytoplanktons means microscopic plants, and zooplankton means microscopic animals. So when they are present in the water, their color will be green, and that gives the sign that there are enough number of phytoplanktons and zooplanktons, which are the food for fish. Next is pH value. So pH value of water must be in between 6.7 to 8.6. If the pH value will be more than this range or less than this range, in both cases the fish production can be less. So if water is acidic, that means if the pH value is less than 6.7, then that water can be treated with lime, which will make the water neutral. The next factor that we should be taking care is dissolved oxygen amount in the water. So oxygen is very much important for the fish. So if it is less, then the fish will start dying. So that is why it has to be in proper amount in the water of lake or pond. And in order to increase the oxygen level in the water, what we can do is we can move the water. We can do the movement of water by stirring. Next is turbidity. So turbidity means transparency of the water. If water has high turbidity, then it will not allow to penetrate light, and because of that the The phytoplanktons which are present in the water or aquatic plants, they will not get sunlight and there will be no photosynthesis taking place. Because of that, there will be less oxygen in this water, which will make death of fish. Similarly, it will also blocks the gills of fish, and because of that, the large number of fish may die. And the last factor that we should be taking care is manuring, because those manure will provide nutrition to aquatic plants, and those aquatic plants can be food for fish, plus aquatic plants can produce oxygen in the water. So in that way, manuring must be done in the pond. Now, what are the advantages of fish farming? Let us discuss. The first thing is that it will provide protein in the form of meat, which is always good for human. The next is the fish oil that is obtained from the fish liver is highly nutritious. Next is the unwanted weeds which are present in the paddy field that can be controlled if we introduce fish in those paddy fields. It also helps to control harmful aquatic insects and larvae. If we release fish into water body, then those fish will consume all those aquatic insects and larvae, and their population can be controlled. And lastly, the there will be self-employment of the people. Because of that, the economic condition of the people will upgrade. With this, I would like to conclude this session. Thank you.